Dear students, I am Devipriya. The last class we deal with the introduction of algae, and in the introduction section we go through almost what about the different types of thallus organization in algae. Now, before starting the class, you all have to identify that algae are major producer of the biosphere. They are the major producer of the biosphere. Since producer means they have to prepare their own food with the help of pigment. The major pigments seen in algae are chlorophyll. In addition to that, there are different types of algae are there. They are Chlorophyceae, Phaeophyceae and Rhodophyceae. That is, Chlorophyceae means green algae, Rhodophyceae means red algae, and Phaeophyceae means brown algae. Another thing is that you already know that the algae are with unicellular to multicellular thallus. Even though they are now also considered it as a plant. This is due to the presence of cellulosic cell wall like plants. Then presence of chloroplast so that it can perform autotrophic nutrition or it behave as producer. Another feature is that the algae shows phototropic nutrition. You all know that due to the presence of this chloroplast. So algae are the major producer of the biosphere, and they are considered as plant due to presence of cellulosic cell wall, chloroplast and phototrophic nutrition. Then we move to the next slide. Today's main section is salient features of algae. Salient features of algae. First, it is heterogeneous in nature. Heterogeneous in nature means the body is made up of different thalloid structures. That is, it is maybe of unicellular to multicellular. So, it is a heterogeneous. Then, 3 levels of organization is noticed. That is, prokaryotic type, mesokaryotic type and eukaryotic types are noticed. So, 3 levels of organization are noticed. Prokaryotes and eukaryotes you already studied from lower class. Then the new term as mesokaryote. Then what is mesokaryote? The cells contain a well-organized nucleus. The presence of well-organized nucleus means it is a eukaryote. But in the case of mesokaryote, what happens? The nucleus divides through a mitosis. That is, nucleus behaves as a prokaryote. So, what is mesokaryote? Mesokaryote cells has well-organized nucleus like that of eukaryote, but its nucleus divides by a mitosis like that of prokaryote. You have to study this for also 1 mark, 2 mark type questions. So coming back, 3 levels of organization: prokaryotic organization, mesokaryotic organization and eukaryotic level of organizations are noticed in algae. Another feature is that the main plant body of algae is haploid in nature. Haploid is the characteristics of lower forms of organism. So the main plant body of the algae is haploid in nature. So we can call it as the gametophyte part or gametophyte. So main part is haploid and gametophyte, or the plant body we see is a gametophyte, or the plant body we see is a haploid type. Then cells is covered by cellulosic cell wall. Like all plants, the algae cell wall is made up of cellulose. Then three classes of pigments. I said in that beginning that there are chlorophyll is the main pigment. Then three classes of pigments are there. They are chlorophylls, carotenoids and phycobilins. Chlorophylls, carotenoids and phycobilins. Then next feature is the reserve food materials. So the algae are of different groups. We have to go through Chlorophyceae, Phaeophyceae and Rhodophyceae with the type study. And before that we have to identify the reserve food materials of algae. In Chlorophyceae, the reserve food material is starch. In Phaeophyceae, the reserve food material is laminarin. And in Rhodophyceae, the reserve food material is Floridian starch. Yes, these all are you have studied in your plus 2 classes and even in 10th standard also. So once again, the reserve food material in green algae is starch, and that of brown algae is laminarin, and that of red algae is Floridian starch. Then another feature is that there is absence of vascular tissue. So vascular structures are not noticed in algae because these algae are very tiny plants with a thallus-like structure. So vascular presence or importance of vascular tissue is not significant. So also there is no vascular tissues are noticed, and also mechanical tissues are absent. Mechanical tissues which give strength to the plant body. Here the plant is or the algae is a very delicate body, and so we can't able to observe any mechanical tissue such as sclerenchyma or collenchyma. Only parenchymatous cells are noticed in algae. So once again, vascular structures and mechanical tissues are completely absent in algae. Then it has hold fast. The thallus has hold fast. Hold fast means it is a special structure which helps the body to attach to the substrate. And it has a stipe which resembles a stem, and it has a lamina which resembles a leaf. So in certain cases, even though the algae is of unicellular to multicellular, even in the multicellular, for the advanced or some unicellular, even some filamentous form has hold fast, and some other have stipe and laminae. So presence of hold fast, stipe and lamina is noticed. Then about the flagella, there will be structure of flagella, the organization of microtubules or the arrangement of microtubules is 9+2. The detail of such arrangement you have to study it later. So the 9+2 type of arrangement of microtubules is noticed in the flagella of algae. Then about reproduction, even though it is a very lower form of plants, it shows vegetative, asexual and sexual reproduction. Then another salient feature is that embryo is not formed after gametic fusion. Here sexual reproduction is there, that means gametic fusion will occur. But even though you know that even though the gametes are fused, what have to be formed, embryo has to be formed. But in algae, even though the gametes are fused, the production of embryo or embryo formation is never observed. So once again, embryo is not formed after gametic fusion. In most cases, the diploid stage in the life cycle is zygote, which undergo meiosis just after its formation. So, in the starting we said that the plant body we see is haploid. So, but in the life cycle we noticed a diploid phase, and that diploid phase which is called zygote, and which undergo meiosis to form again the haploid cells. Then sporophytic, another feature is that the sporophytic and gametophytic generations are independent. Sporophytic and gametophytic generations are independent. That means sporophytic generation means diploid generation, and gametophytic generation means haploid plant body generation. They are independent because they are independent means the plant body we see is a gametophyte. From the gametophyte what will be formed? Sporophyte will be. From the gametophyte, sporophyte or diploid stage is formed. Even though the sporophyte and gametophytic phases are noticed in algae, the plant body we see is the gametophyte, and from the gametophyte sporophyte is formed. But that sporophyte is independent of the gametophyte, or we can say in another sentence that the gametophyte or no, the sporophyte is not is the sporophyte never be a parasite on gametophyte. So sporophytic and gametophytic generation are independent. So no parasitic sporophyte on gametophyte. Once again, the plant body we see is the gametophyte. From the gametophyte, what will be formed? Zygote will be formed, and that zygote is the only sporophytic phase of the life cycle of algae, and that sporophytic phase is independent, or it will never act as a parasite on gametophytic plant body. And these all are the salient features of algae. Then coming to the topic, there before in the salient features we have said that there is different types of reproduction is there. That is reproduction by vegetative means, asexual means and sexual means. It may be asked, from the salient features itself may ask for essay type questions. On going through the different type study, we can elaborate the writings, and then another essay type question may be asked on the basis of reproduction. So here the reproduction is vegetative, asexual and sexual. Here we are going through vegetative reproduction. There are binary fission, fragmentation, amylum stars, hormogonia, bulbils and achaenids. And there are so many other type of vegetative reproduction are there. We have to go through binary fission, fragmentation, amylum stars, hormogonia, bulbils and achaenids. Coming to binary fission, it is the simplest method of reproduction. It is the simplest method. Unicellular forms reproduce by this. So, binary fission means simply a cell divides into two. Here, the Chlamydomonas is the best example to cite for binary fission. So, Binary fission means simple method of reproduction, and unicellular forms are usually unicellular algae shows this binary fission type of vegetative reproduction. Then about fragmentation, you all know that fragmentation. What is fragmentation? Fragments are breaks into many fragments, or the filaments are breaks into many fragments. Each of that fragments can give a new organism. Example: Spirogera, Eulothrix, Eudogonium, Cycnema. They all are filament types. The Spirogera, Eulothrix, Eudogonium are filamentous algae, and that filaments can break into several fragments and gives new filaments. Then comes the amylum stars. It is a star-shaped aggregation of starch which contains cells and which may develop into new organism. And usually this type of star-shaped structure is noticed in algae which named Chara, Chara, Chara. So star-shaped aggregation of starch containing cells which may grow into a new organism or a new algae, that is called amylum stars. Example: amylum stars is noted in Chara, Chara, Chara. Then Hormogonia. Hormogonia means it is a filament like body, or the cells may break into many celled segments, or the cells may break into many celled segments, and which is covered by a sheath. Many celled segments which is covered by a sheath, and they remain delimited by the formation of heterosis. That means it may be separated from other cells, and it forms, or it may be formed by the death and decay of intercalary cells of the trichome. Trichome means a filament-like structure, and within that filament-like structure, cells may divide continuously to form a rounded structure. And what happened, that cells may get separated, or that cells may separated from the nearby cells, and or by the forms a heterosis may be formed, and by the death and decay of the intercalary cells, what will be developed? Hormonia will be released, and that hormogonia may be develops into a new algae. For example, it is noted in nostoc and oscillatorium. Then another one is bulbils. Bulbils is noted in bulbil, small structure. It is a tuber-like outgrowth developed due to storage of food at the tip of rhizoids and on the lower node of Chara. And it is a tuber-like structure which is noticed, and the bulbil formation is noticed again in Chara, and is found at the tip of the rhizoids. Tip of the rhizoids. Rhizoids means root-like structures. Root-like structures, and such tuber-like structures we can call it as a bulbil. Another one is achainids. Achainid is non-motile dormant cells. Achainid means it is a non-motile, it can't able to move. It is a dormant cell formed by the filamentous heterosis forming cyanobacteria. For example, nostoc. And the formation of achainid is noted in nostoc, and it is a dormant cells, and the dormant cells which is seen in the filaments, and the dormant cells can again develops into new algae during the favorable season. And such so that a single organism can be developed into numerous algae or numerous nostocs. So here we go through different types of vegetative reproduction. They are binary fission and fragmentation, amylum stars, hormogonia, bulbils and achaenids. Then comes the asexual reproduction. Among the asexual reproduction, there are two types. One is of motile spores and another of non-motile spores. So in the under asexual reproduction, there is only one motile spores are noted. We can call it as zoospores. Motile spores means it has two flagella is noted. An example, it is noted in Eulothrix and Ectocarpus. So asexual reproduction or the zoospores are motile spores formed for asexual reproduction in Eulothrix and Ectocarpus. Then comes the asexual reproduction types with the non-motile spores. They are different types. One is aplanospores, another one is hypnospores, third one is exospores, endospores, autospores, carpospores, heterospores, etc. So many different types of spores. I think that you already hear about exospores and endospores, isn't it? So we have to go through aplanospores. Then what is aplanospores? Aplanospores are thin-walled spores, and again we can say that it is a non-motile spore because it has no flagella. So it is a thin-walled spores formed in the vegetative body of algae is called aplanospores. These spores can release from the body and can be developed into a new organism. Then is hypnospores. Hypnospores is not differ from aplanospores. It is same as that of aplanospores. What is the difference is that these hypnospores are thick-walled spores. Both the aplanospores and hypnospores are non-motile spores, and these non-motile spores, hypnospores, are thick-walled aplanospores. The examples, it is in Vaucheria and also in Eulothrix. Then next one is exospores and endospores. The difference is that if the spores is formed within the cell, then we can call it as endospores, or if the spores are formed as a projections from the body, then we can call it as an exospores. So in certain cases, the body may show, algae may show formation of exospores or endospores, or these spores are released from the body or released from the thallus can develop into a new organism. Another one is carpospore and autospores. The one difference is that in carpospore is a diploid spore and it is produced in red algae. So carpospore is a diploid spore produced by red algae. Example, Polysiphonia. Polysiphonia is an example for your type study in red algae. So in studying the type Polysiphonia, we go through in details how these carpospores are formed. So another one is autospores. So autospore is a non-motile spore. Non-motile means it has no flagella, produced within a parent cell and is the same shape as the parent cell before release. And that example is chlorella. So within the chlorella cell, what will be formed? A spore will be formed, and that spore has same size as that of the parent cell. In that stage, it will be released from the parent cell. Such spores are called autospores. It is also a non-motile spore. Then another one is tetraspores. Tetraspores, in some algae, they are diploid. In such cases, what will be formed? Haploid aplanospores will be formed, which is called tetraspores. And these tetraspores are formed in the tetrasporangium. And this formation of tetraspores is again in Polysiphonia. Before that I have said that carpospores are formed, and these carpospores and tetraspores are the special features of Polysiphonia. Polysiphonia is a diploid algae which produce haploid aplanospores which is called tetraspores. Diploid nucleus of the tetrasporangium divides meiotically to form 4 haploid nucleus which develops into 4 tetraspores, and the tetraspores can germinate into male and female gametophyte. So while studying the life cycle of Polysiphonia, we can go through in detail. So we can understand what is tetraspore as. It is a aplanospores, or it is a haploid aplanospores which is produced in the Polysiphonia within the tetrasporangium. Then comes the last section, sexual reproduction. So sexual reproduction, there are 4 types of sexual reproduction. I am sure that you have studied about 5 types. You have studied about 3 types: isogamy, anisogamy and oogamy. But before going to isogamy, anisogamy and oogamy, you have to know about the lower forms of sexual reproduction. They are autogamy and hologamy. So in autogamy, the fusing gametes are fused and developed from the same mother cells. The gametes are fused, but what is the negative is that the gametes are formed from the same mother cell. So there will be fusion or the zygote is formed, but no new characters will be developed. So fusion occurs, but that cells are formed from the same mother cells or from the same plant. So what will be, there will be no fusion of characters occur. So such sexual reproductions are autogamy and is noticed in diatoms. Another sexual reproduction lower form shows hologamy. Hologamy means vegetative cells of different strains, and that strains we can said as plus strains and minus strain, and they behave as gametes, and after fusion they form zygote. New genetic combinations are developed here, but what will be there? There will not be any multiplication, because plus strains and minus strains comes together and there will be some exchange of genetic characters, but no new individuals will be formed. There is fusion occurs. And example for hologamy is chlamydomonas. So what's the difference between autogamy and a hologamy? In autogamy, fusion occurs and zygote form, but no new characters will be formed. In hologamy, again fusion occurs, no new individual formed, but fusion of characters occurs or new characters developed, but no new individual formed. Then isogamy. It is the process of union between two gametes which are morphologically and physiologically similar. After fusion, they can form a zygote, or they form a zygote. That gametes are called isogametes. Usually they are flagellated. So isogamy means fusion between two gametes, and that two gametes are similar. On what basis? Morphological basis and on physiological basis, that two gametes are similar, and such gametes we can call it as isogametes. Example, Chlamydomonas, Ulothrix, etc. The picture depicts this isogamy. These two gametes are similar. It will be fused to form the zygote. Then anisogamy. There is some dissimilarity. In this process, the gametes are morphologically and physiologically different. The smaller and more active one is the male, whereas larger and less active one is the macrogametes or female. This anisogamy is noticed in Chlamydomonas. While studying anisogamy, you have to know about another term, physiological anisogamy. Physiological anisogamy means gametes are morphologically similar but physiologically they are different. Morphologically they are similar but physiologically different. Usually anisogamy means morphologically and physiologically different. Physiological anisogamy means only physiological difference is noted. On morphologically they are similar. The two gametes are similar. Examples Cycnema, Spirogera are the two examples which show physiological anisogamy. The picture of physiological anisogamy, one is large macrogamete, another is small microgamete and they fuse to form zygote. The last one is the oogamy. It is the advanced process where fertilization takes place between small motile male gamete with a large non-motile female gametes. So male gamete is small and motile while female gamete is large and non-motile. The male gamete we can call it as sperm or anthrozooids and female gamete we can call it as egg or ovum. Male gametes usually developed within the antheridium whereas female gametes develop within the oogonium. So these two terms you have to familiar with, antheridium and oogonium. The example for algae which shows this oogamy is Eudagonium, Vaucheria, Chara, Sargassum, Polysiphonia. These all are for your type study in this semester under phycology. Eudagonium, Vaucheria, Chara, Sargassum, Polysiphonia, etc. The all algae shows oogamous type of fertilization. Here the picture, this large one is the egg and small one microgamete is the sperm which fuses to form the zygote. That is the oogamy. Dear students, now I am winding up. Today we deal with the salient features of algae and also the different types of reproduction noted in algae. While studying details with each type, we go through different types of what are the different types of reproduction noticed in that particular type. So students, hope you are safe at home and you have to study or utilize the time for studying the topic. You also prepare note for what I am teaching during this lockdown period. So winding up, thank you.